Good evening, good afternoon, depending on where you are in the world today. Thank you for joining us. First, I'm going to spend about 10 minutes kind of as a primer for Aaron's talk, giving kind of a general introduction to the recent improvements we've made in the Smart Link in the past couple months. So for those of you who are new to PacBio analysis, we have a software suite of tools called Smart Link that allow you to analyze, visualize, and manage your data. So it's good for both the wet lab and the bioinformatics. So there's some wet lab stuff for sample setup, the run design on the machine, QC, and then there's some data management. So beyond just the data you're sequencing, but also say reference genomes you want to align to or barcode files. And then smart analysis, which of course is the secondary applications that we provide. This is kind of nice because we also provide this, like this example you saw was a GUI example, whereas we also offer this as a command line or an API for the more bioinformatics people. So updates for V11, we've got several big things. I would say that one of the big take-home messages is the old version still had a little bit of the old types of data lying around, and now we've really gone hi-fi centric. So all the applications now in this are now hi-fi based. So basically we're getting very long and very accurate reads now, so we can use those for our analysis. More of this is actually done now on the instrument. I'll go through that. We have new applications such as AAV sequencing, and I won't go into this much because this will be Aaron's focus today is on 5-base sequencing, basically that 5MC sequencing that we now support. Some changes for those of you who are using earlier versions. Now we've moved, as I said, to being hi-fi centric. So really the raw data that you're kind of getting now off is going to be that hi-fi data. So we've moved from what used to be called reads.bam. So reads.bam also have lower quality CCS data and some other types of reads. Now we've just, the raw data you're going to get is a hi-fi reads.bam. So really just hi-fi data. For those of you who are new, a hi-fi is a CCS read with a cutoff of QV20. And then there's some, of course, some optional files. So for instance, if you use Aaron's new, what he's going to present today, the 5MC, you'll also get these 5MC files. Again, this is just running this home. Why do we switch to hi-fi reads only as our default? Well, one is it's about 50% smaller file size now to switch from reads.bam to hi-fi reads.bam. Plus, it just makes things much simpler because I think most of us are really only using the hi-fi reads anyway for our analysis. So you don't have to now, the very first step of your analysis, filter out the non-hi-fi data. However, if you have upgraded to V11, you can still get, for instance, the reads.bam that contain the low quality CCS. That's an option even in the GUI. However, that's incompatible with the on-instrument 5MC calling heteroduplex detection demultiplexing that I'll present in a second. And if you still want to get the sub-read data, that's also still possible with some workarounds that we can provide. So these are those main things that you have in Smart Link. So some of the key features that has changed for V11. I won't go on this today because it's more for the wet lab, and this is a bioinformatics focus series. But the sample setup has a new high throughput mode. The run design has new options. There's new run QC plots, and smart analysis has changed a bit. So like I said, I'll go through most of this. I'll just skip a little bit of the wet lab parts. So for the run design, one thing that's also now done is the kinetics, the CCS kinetics, we're only keeping for when you want to do the microbial analysis. For other types of run designs, that's actually turned off now by default. The reason for that is basically you get 5 times the file size with those kinetics. But again, that's just the default. You can still turn that on if you wish. Applications, most of it looks like what you had before. You still have whole genome sequencing for hi-fi or microbial, isoseq, viral sequencing, metagenomics for 16S or the shotgun methods, and both the shorter and longer amplicons. But now we also have this new adeno-associated virus application. And when you click on these applications now, you also get this kind of auto-filled selection of the various fields with highlights in green for the default values, which you can of course change. So these additional on-instrument options. So the four things we really have now is we have heteroduplex detection. So you get a heteroduplex when this is the one strand of DNA and purple being the other strand, is if these don't match up perfectly. So this could be due to similar molecules annealing to each other, or it could be due to, for instance, if you ran a PCR and the polymerase during the PCR had a small hiccup and incorporated the wrong nucleotide on one strand. So this is important for applications like amplicon sequencing or the AAV I mentioned, but now we can actually sort those out now by basically making a consensus from each strand if you want to. Moving on to the AAV. So first of all, just a quick nod. This webinar series will be happening every 2 to 3 months. And just a heads up, in November, this is actually going to be AAV focused. AAV is a little bit different than standard sequencing. So normally you have an adapter always on both ends. But for AAV, if you have a self-complementary structure, so this is what you're seeing here, we end up only with an adapter on one end. And that causes it a bit of difficulty in our adapter calling. So basically, we used to have to do this pipeline for recalling adapters. It was annoying, it took time. Now, if you set up AAV and run design, that's all taken care of for you. And it also includes the heteroduplex detection. We also have on-instrument demultiplexing, so you can save yourself some time and not have to do that downstream on your own platform. Again, this can be on instrument. If you have a Sequel IIe, this can be done in SMRT Link or not done. One heads up though is if you do this, the undemultiplex hi-fi reads.bam file is not transferred. However, for you bioinformaticians that are command line savvy, you can still use Lima to remerge these demultiplex files if you ever needed. And finally, again, the main talk for today that Aaron will discuss is the 5MC calling. This is just a preview where we see a bunch of reads here, and you can already see here highlighted in red or methylated positions and in blue unmethylated positions in these reads. So you can really see these stacking up at certain parts, but I'll leave that for Aaron. Run QC, there's been a couple updates too. In the past, we had other types of reads besides hi-fi in a lot of these plots. So these are now, as I mentioned, it's all hi-fi centric. So for instance, this is a new version 11 plot of your read length distribution. It's all just one type of read, it's hi-fi data. Whereas in the past, we had both hi-fi reads, non-hi-fi CCS data, and other data. Same thing goes, for instance, for your quality distribution. Now it's only the hi-fi data, so hence you see this cutoff of Q20 right here, whereas before it went below Q20. And same thing when you want to plot the read length versus the predicted accuracy. Now you can see that it's all huddled around here because this is all hi-fi data, whereas it wasn't before. And in terms of how smart analysis, the secondary applications, there's been some updates. You can still do your analysis, or you can set it up for auto analysis. So as soon as it's done sequencing, it starts to run. But we've also separated now between analysis and data utilities. So an analysis you can think of as more of something that's a pipeline for to produce a biological meaningful result. And these accept only hi-fi reads. Whereas we also offer now some data utilities, which are kind of some of those little small jobs that basically just help you manipulate the data. And these in general accept hi-fi reads, though we have one option that does not, which I will show in a second. So for the analysis applications, we've got a lot of the ones you had before. We have genome assembly that stayed the same, hi-fi mapping, the viral sequencing. IsoSeq has changed a bit. It used to use a cutoff of Q10 and now uses a cutoff of Q20. So it's also hi-fi reads. It's all standardized now. And we've also done a merging of what used to be a microbial genome assembly and a separate base mod. That's all now in one. So you can do both microbial assembly and base mod analysis in one tool. And then we have minor variant analysis and structural variant calling. And then, like I said, we've kind of moved these over to these data utilities. These are these kind of small things that, just for data manipulation. So for instance, the 5MC, demultiplexing, export reads, which is also a way of, besides just changing read formats, putting cutoffs. We have options. So for instance, if you used a low input sample, we have the mark PCR duplicates and trim ultra low adapters. And this is the one exception that does not run on hi-fi reads. We still offer the circular consensus sequencing module, which is also where you can put your cutoffs, for instance, to make a hi-fi read. And this is just the last thing, as I mentioned, is microbial genome analysis. This has changed a bit. It used to be 2 modules. Now it's all 1. So you can get your basically de novo assembly of small genomes and companion plasmids, as well as identify the methylated bases and associated nucleotide motifs, and optionally also identify 6MA and 4MC modified bases and associated motifs. And similarly, some of these plots that used to have other types of data, this is all HiFi based now. So notice that the plots have changed a bit. So a bit of take-home message before I hand it over to Aaron for the main talk today is for SmartLink v11, we've definitely moved everything to be HiFi centric. Basically, it helps you, it's less data, it's making their file size, not less data, smaller file sizes, and it just makes things much simpler now that it's all been standardized. And it's great now you have all these additional on-instrument options for demultiplexing, heteroduplex detection, AAV applications, and the 5MC calling. And again, we've now simplified where you can do your microbial genome assembly and base calling in one analysis.
So thanks everyone for joining today. As Matt said, I'm going to talk about PacBio's new Firebase HiFi sequencing approach. Just as a higher level view of what we develop at the company, we have a series of technologies aimed at providing the most comprehensive and accurate view of genomes and epigenomes. Those include the nanobind technology for DNA extraction, the HiFi sequencing technology that we are talking about today, and also an in-development short-read sequencing technology focused on or built around the technology of sequencing by binding. In terms of the HiFi sequencing technology, it's a platform that produces DNA sequencing reads that are both long and accurate over the standard 4-base DNA alphabet that we usually think of the ACs, Gs, and Ts. The HiFi reads are around 15 to 25,000 bases long, and the typical accuracy of a read is greater than 99.9%. So these are reads that have extremely high accuracy and are quite long. And this type of read has been shown to provide the most comprehensive view of genomes. It was the basis of the first telomere-to-telomere assembly of a human genome, and it reveals additional variation in genomes and other technologies. So you get a complete view of structural variation in addition to single nucleotide variation in indels. And further, it's been shown that this more complete view of the genome is important for biology, and in particular, this is one example of looking at around 400 HiFi genomes in a rare disease cohort and identifying a number of new explanations for rare disease by incorporating structural variations detected with the HiFi reads. But of course, for phenotype, it's not just the genome sequence that matters. There's also the epigenome, in particular DNA methylation, that contributes to phenotype. For humans and other vertebrates, the DNA modification, the epigenetic modification that is most prevalent is 5MC at CpG sites. So the addition of a methyl group at position 5 on the cytosine bases, and this epigenetic modification is typically associated with silencing of the corresponding DNA fragment. So it's the functions of DNA methylation include silencing damaging DNA elements like transposons. It's also key to maintaining cell identity, where daughter cells inherit the epigenetics of their parent cell and thus have the same cell identity. It's also key to X inactivation for dosage compensation in females. And DNA sequencing technologies can be used to look at this modification in bases. In particular, for short read sequencing, the typical approach is to use bisulfite conversion. So here, if you wanted to determine the genetics and epigenetic profile of a certain DNA library using short read sequencing, you'd first sequence the whole genome sequencing library to get the sequence, and then you could do bisulfite conversion of the DNA to convert non-modified Cs into T bases, and methylated Cs will stay as Cs. So you can do bisulfite conversion, then whole genome bisulfite sequencing to determine the epigenome. It's quite different with HiFi sequencing, where there's no bisulfite conversion, there's no second set. It's just a single standard DNA sequencing library, and you get directly from the instrument the Acs, Gs, and Ts, and the 5MC. And this is possible because of the fundamental properties of PacBio sequencing, Well, one of which is that it's sequencing native DNA molecules. So there's no amplification or any other modification to the native DNA in library prep. You just add adapters on the end, but it is the original DNA molecules that are being observed by a single, it's a single polymerase sequencing a single DNA molecule that's being observed. And that's one key property that the epigenetic modifications are carried through to the sequencer. The second is that the signal that you get from PacBio sequencing is the observation of fluorescently labeled nucleotides being incorporated into a DNA strand, and the different fluorescent labels indicate the base identities. But in addition to seeing the base identities, you also observe in real time how fast the polymerase is moving, how long it takes to incorporate a base, how long it takes to go from one base to the next. And these are referred to as the pulse width, which is how long a base takes to be added. And then the interpulse duration, which is how long it is until the next base starts to be added. So this pulse width and interpulse duration, the so-called polymerase kinetics, are impacted by epigenetic modifications like 5MC. In particular, DNA methylation tends to slow the polymerase as there's an additional obstruction that the polymerase has to move past. The kinetics are represented in the kind of raw calls you get from the PacBio sequencer, the subreads.bam format. That will show the bases that are called. So in this example, it showed G, C, T, C, and G. And then there's two additional tags, the PW tag, which stores the pulse width, and the IP tag, which stores the interpulse duration. And these are just stored in whole numbers. These are counts of camera frames, because this signal is taken on a camera that on the Sequel II system operates at 100 frames a second, observing the kind of intensity of fluorescence, and so that the integer number of frames that are spent in a pulse or between pulses are encoded in these PW and IP tags. The interpulse duration assigned to a base is how long it takes prior to that base. So for example, this C base here, the interpulse duration for that is the amount of time between the preceding C, or the preceding G and that C. Now with Hypo sequencing, you don't just get a single subread, you observe the same molecule sequenced multiple times in series and get a number of subreads that are combined in a consensus software to produce the accurate Hi-Fi read. But the CCS software, in addition to producing the consensus sequence, also produces consensus kinetic values. And the way that works is you could, just as an illustration here, you could look at this one position in the molecule, and it has here showing 8 different subreads, 4 from the forward strand and 4 from the reverse strand. And each of those is a different subread, and for each of those, you have a pulse width and an IPV. So you can envision these subreads aligned to the Hi-Fi read, and the bases that align to that Hi-Fi read, you have the recorded pulse width and IPV for those corresponding bases. Then separately for the forward strand and the reverse strand, since they are 2 separate DNA strands, they can have separate kinetic, separate methylation status. And so separately for the forward strand and the reverse strand, you have all the pulse widths and IPDs, and what CCS software will compute is an average of the pulse widths and IPDs for a given strand. The final Hi-Fi reads, then they have the base sequence, and then they have these 4 summary kinetics values. The average pulse width on the forward strand, or the FP tag, the average IPD on the forward strand, the FI, and then similarly for the reverse strand, RP and RI. And the forward and reverse is just based on whichever strand was selected to output the Hi-Fi read. So it's typically the first strand that was observed. So every Hi-Fi read will have these kind of summary kinetics values, and because different DNA epigenetic modifications have different impacts on kinetics, you can then look at those average kinetics and figure out whether a base is modified and what type of modification is present. So this is just showing two different epigenetic modifications. One is the addition of a methylation on A bases, Here, this is on the x-axis, or this is showing an artificial DNA template that has 2 methylated positions shown by the red triangles. And the y-axis shows the interpulse duration and how much it is different from what you would expect. And what you can see for 6MA is that you get a very big impact on the interpulse duration directly at the modified base and also a few bases downstream. For 5MC, this is again the same type of setup. You have an artificial template. There are 2 modified bases, denoted by the triangles here, and you can see that there is a perturbation of the interpulse duration, but it is not as focused as 6MA, and it's a bit more dependent on the other DNA-based context. But nevertheless, there's a distinct signature that denotes 5MC versus 6MA, and we can train models that pick up this signal and call the modification or the probability of modification. And that's precisely what we did for 5-base DNA sequencing. So the 5-base DNA sequencing is built on a neural network that runs after CCS on hi-fi reads. So you first call the consensus sequence and the consensus kinetics, and then a neural network model is run over that sequence to assign to every CPG site in the read a probability of methylation. And the model is a fairly simple convolutional neural network. It starts with the input to that model is the sequence around a CPG site, plus or minus 8 bases, and the forward and reverse interpulse durations and pulse widths. So you get the kinetics values for both strands. Apologies. You get the kinetics values for both strands, the DNA sequence, and the number of passes. The model is trained to call a single probability of methylation for a CPG site. So there's an assumption in the model that the 2 C's here in the CPG, the C on one strand and C on the other strand, have the same methylation status, which is typically the case in human CPG methylation. So you're getting a single call for each CPG site out of this network. The network is trained on a set of positive and negative controls. So the way it learns what is the kinetic signature of a methylated CPG is it's trained on a whole genome amplified human data set where there are no epigenetic modifications as they're removed by the PCR and the amplification. So that's the negative control set. And then there's a positive set, which is the amplified DNA treated with methyltransferase that very efficiently adds 5MC to all CPG sites. It uses MSSS1 enzyme that will very efficiently add methylation. So that gives a set that is 100% methylated. So that's the kind of setup for the network. It's this looking at a relatively small window around each CPG site, assuming symmetry of methylation status, and trained on that positive and negative set. And so you pass into the model a vector of these kinetics, and you get out the probability of methylation. This model is integrated onto the Sequel IIe system. So you actually get directly on runs from the Sequel IIe system a hi-fi reads.bam file that has the sequence and quality values that also has the methylation. The methylation, I'll talk about the encoding in a second, but the methylation data adds a fairly small amount to the file size. So you have about a 30 gigabyte file size for hi-fi reads BAM, and you get around another 5%. So something like a gigabyte or 2 to encode the methylation information. The compute for this occurs on the system. It's also able to be run in SMRT-Link, as Matt had mentioned, but it does come as part of a standard Sequel IIe run. It's run by default. The representation that is used to encode the methylation information is a standard MM and ML tags, which are part of the SAM-BAM specification. And I will go into some more details in that in a second. And this tag, because it is standard, it's not a PacBio specific tag, it's well supported in IGB and other tools. Okay, great. So to give a sense of the processing, I mentioned before you can run this either on or off instrument. If you want to run it off instrument, you have to do what Matt had mentioned earlier, which is to retain the kinetics information. And then you can run this binary primrose, which will accept a BAM that has these summary kinetics tags, and it will output a BAM that has the methylation information. This is also able to be run on instrument, so this whole pipeline can run on instrument, or will run on instrument by default. To give a sense of how to interpret these tags, so this MM and ML tag, which are the SAM BAM standard way of representing methylation information, they're a bit. It seems that it's a bunch of numbers, a bit hard to read for a human, but it's actually a fairly simple encoding. So this MM tag, it describes which mark is being described. So here it's C plus M, which is the code for 5-methyl cytosine. So the M for the 5-MC modification, the C being the bases that are affected, and plus telling it that we're describing this on the forward strand of the read. Then there's a series of numbers, and that tells you how many C's to skip before you're in between ones that are being tagged. So here we're only annotating CPG sites. So if you look at this read, there's a number of C bases in that read, and the first CPG site would be here, and you would skip bases 1, 2, 3, and 4. So you skip 4 Cs, and then you annotate a CPG, and then you skip 1 C, and you annotate the next CPG. So it's just a way of indexing which C you're describing in the read. And then the probability of methylation is encoded. So there's a 1-to-1 correlation between the MM and ML, and the probability of methylation is encoded as an integer with an implicit denominator of 255. So this would say this first CPG has a probability of methylation of 249 over 255. So high numbers are very likely methylated, low numbers are unlikely to be methylated. When you look at that in a genome browser, it's typically shown where low probability methylation is shown in blue and high probability methylation is shown in red. So kind of the raw information out of the models is for every single CPG in a hi-fi read, you get this probability of methylation. The reads are all treated independently. It's only a read level call. But then you can take these reads and look at a pileup and think of calling the percent of bases at a given position that are modified. And for that, we have a second software tool called PBCPG tools. So to look at these two analysis software tools, Primrose, which is the methylation caller, its input is the hi-fi reads BAM with kinetics, and its goal is to call the probability of 5-methyl cytosine at every CPG site. It looks at a window of plus or minus 8 base pairs around the CPG site and it assumes symmetry, and it outputs a BAM with the MM and ML tags per read. This is processing individual reads. And then PBCPG tools, its input is a pileup, so aligned hi-fi reads.bam to a reference. Its goal is to estimate the fraction of reads methylated at a given position. It looks at a window of plus or minus 5 CPG sites around a given CPG site, and it will output a big WIG file that has, for every single CPG position in the reference, the fraction of reads that are methylated. Okay, so that's the basic analysis tools to give a sense of how the data look and what are the benefits of 5-base hi-fi sequencing. So some of the key benefits are that it's a genome-wide measure of methylation. So it comes just with the standard hi-fi reads, and those hi-fi reads in a whole genome sequencing library are covering the whole genome very uniformly with a high ability to map into even difficult regions. The other key advantage is that this comes from a single library and a single sequencing run. There's no additional sequencing that has to be done to measure methylation. And then it comes with the standard hi-fi reads. So these are long reads. They can be phased into haplotypes, which means you can also phase the methylation. You get uniform coverage of the genome, and you get high mapability, so you can access much more of the genome than you can with bisulfite sequencing. At a gross level, what you can look at is for a human cell line here, HG002 differentiated lymphoblastoid cell line, what are the probability of methylation calls across all CPGs in all reads? You can see most positions are methylated, which is what's expected for humans. So you see this kind of bimodal distribution where most bases are either very likely not to be methylated or very likely to be methylated, and a bias towards methylation. Interestingly, if you look at other human cell lines, in particular, if you look at the CHM13 cell line, which was the one used for the telomere to telomere, the first complete human genome assembly, that is from an undifferentiated trophoblast lineage, and it has a much different methylation profile. So at that undifferentiated stage, many positions are unmethylated. So you can see even in the HiFi data at the broad level, the different epigenetic characteristics of a differentiated cell line from an undifferentiated one. The second thing you can look at is the accuracy of these calls, and if you compare the percent methylated by position that we see with HiFi reads to what you see with other technologies, you see very high concordance. So here we're comparing HiFi reads to a number of other different approaches to measuring 5-methylcytosine. So there's two different methods for EM-seq and whole genome bisulfite sequencing. This is an enzymatic modification approach with short read sequencing. This is bisulfite modification with short read sequencing, and then two other long read sequencing approaches with nanopore sequencing. You see this is measured for 3 different humans, all these different approaches, and you see high correlation between the methylation calls you get from HiFi sequencing and these other approaches, and much higher correlation within an individual across technologies than for technology across individuals. So I think this is a good indication that what's being measured here is not a bias of any single measuring approach. So you don't see, for example, that HiFi for HG2 looks more like HiFi for HG1 than it does the MC. So that we're measuring the biology of these samples and not the methods. Further, we see that with HiFi sequencing, the correlation between the methylation calls you get with HiFi and what you get with whole genome bisulfite are quite robust to coverage. The data that I showed on the prior slide was the correlation for HiFi at high coverage around 50x HiFi sequencing, which is very high. But you see as you go down to lower coverages, even down to 5x coverage, that you see a good correlation between what you see with HiFi sequencing and what you see with bisulfite sequencing. Well, now in terms of how the data look biologically, you can look at a number of different interesting characteristics. So this is for a human cell line, HG002, that differentiated lymphoblastoid cell line. And looking at here about a 70,000 base pair region of the human genome, just a representative selected region of the genome with a few genes. And you see most of the genome is red, it's methylated as expected. And there's a few columns here where all the reads are showing hypomethylation, and those correspond to the transcription start sites in this region. So that's an expected biological observation, that you see reduced methylation at active transcription start sites. So far I've been talking all about human, and the model was trained on treated and untreated human DNA, but the model generalizes well to other species and other situations. So this is just showing that same orthologous locus in chicken, and you can see again that you see overall high methylation, except for at these transcription start sites you see columns of reduced methylation. So looking at a second biological observation is X inactivation. So one biological role of 5-methylcytosine in humans is to silence is part of silencing half of X chromosomes in females for dosage compensation. And you can see that nicely in this example, which is that same HG002 cell line I've shown previously, and now 2 other cell lines, one derived from the father and one derived from the mother of that individual. And this is just a representative region of chromosome X. So here the son and father just have 1 copy of X, and the mother, of course, has 2 copies. And if you look at the gene transcription start sites, you see in the males that all of the reads are hypomethylated, meaning all of the chromosomes are active. While for the mother, you see that half of the reads are red and half of the reads are blue at the transcription start sites, so showing half of the chromosomes are silenced and half of them are active. One other interesting aspect you get of having methylation as part of these long accurate reads is you can get haplotype phasing of the methylation. So you can look at this same human trio that I showed on the previous slide, HG2, 3, and 4. and Hi-Fi sequencing of a particular region in all 3 individuals. So you see long accurate reads. Within an individual, you can break those into haplotype by the single nucleotide variant differences. So you see really 2 different alleles in each individual. And because it's a trio, you can ask for the son which allele was inherited from the mother, which was inherited from the father. So in this case, the top allele in the son matches the top allele in the father, and the bottom allele in the son matches the top allele in the mother. So this allele is paternally inherited, and this one is maternally inherited. Now these reads are picked from this locus, which is paternally expressed gene number 3. It's a parentally imprinted gene. So this is a region where, although it's an autosomal region, so although everyone carries 2 copies, the only one that is active is the one that's inherited from the father. And you can see that here, that the paternally inherited copy has low methylation, is blue, and the maternally inherited copy is red, is silenced. So here you get this combination of accurate reads allowing phasing, the trio allowing assignment of inheritance, and then that being consistent with the methylation. It's interesting to, and I think that nice to separate the ability of the Firebase sequencing to find the genetic sequence and the epigenetics, to look at the same region in mouse. So this is Hi-Fi sequencing of the laboratory black 6 mouse. So it's inbred, there's no genetic variation. And this is for that same paternally expressed gene number 3, which is also an imprinted gene in mouse. So you can see in mouse, there's no genetic variation because it's an inbred strain. But nevertheless, the mouse that was sequenced still had a mother and a father. And so although there were genetically identical mother and father, but the imprinting is still, since you can see half of the reads are blue and half of the reads are red. So showing that the Hi-Fi sequencing is able to here find the epigenetic signature completely separate from the genetics, because all these reads have the same sequence, but they do have different epigenetic profiles. Another advantage of the Hi-Fi reads is that they're able to map into some really difficult regions, including into the most repetitive regions in human, the centromeres. So this is just showing now at a very coarse scale, megabase scale, the average methylation as seen with Hi-Fi sequencing in the chromosome 3 and chromosome 19 centromere. And you see these interesting profiles of some regions showing fairly high levels of methylation, some showing paucity of methylation. You see these dips, which are part of the centromere biology, and they associate with the kinetic core, where the pairs of chromosomes will come together. So the profile you see with Hi-Fi sequencing is very consistent with the profile that has been previously published. And not only the shape of these profiles are consistent, but also the percent methylation. So you see here the percent of reads that are methylated are somewhere around 65%, which is exactly what you see with other methods. So they see good consistency even in these very difficult regions between, in this case, Hi-Fi sequencing on the top, and this is nanopore sequencing on the bottom. So those are the key benefits of Hi-Fi sequencing, and just to show some examples of how it can be applied. One example in rare disease is from Matthew Bainbridge at Rady Children's Institute, and he sequenced an individual that had a diagnosis of Prader-Willi syndrome. And here, this is showing a particular region on chromosome 15, but the Prader-Willi syndrome is caused by a biallelic loss of expression of key genes on chromosome 15. It's typically caused by a deletion of the paternal allele. So you have and the maternal allele is silenced by genetic imprinting. So you have individuals where the maternal copy is turned off epigenetically and the paternal copy is absent. But in this individual, the individual had a regular copy number of chromosome 15. They did not have a paternal deletion. And further, they had the normal heterozygosity on chromosome 15. So it wasn't a case where you had like 2 identical chromosome 15s. So from Hi-Fi sequencing, you could see that this individual had 2 chromosome 15 copies. You could separate the reads into haplotype 1 and haplotype 2, but they were both methylated. And so you can compare here the these are showing probabilities of methylation, so it's very high for both copies in this individual, and these are just 3 control individuals, where you see, again, 2 haplotypes. One of them is silenced and one of them is on. And it turns out in this case, this individual had uniparental heterodiacomy. So this individual had inherited 2 chromosome 15s, but both of those came from the mother. So it was likely an egg that had 2 chromosome 15s that was fertilized by a sperm that also had a chromosome 15. The cell rejected one of those chromosome 15s, but threw away the paternal one, leaving the 2 maternal copies. So they are different chromosomes, but they have the same epigenetic status because they're both maternally inherited. So it's an interesting case where you can use this HiFi sequencing to see the full picture, the copy number, the phasing, and the methylation status. Another interesting application of the Firebase HiFi sequencing is to look at repeat expansions. So repeat expansions are regions of the genome where there's a small motif that is, so say, the repeat expansions are tandem repeats present in the genome that increase in copy numbers. You have, in this case, the FMR1 repeat is a region of the genome where there's a CGG repeat near the FMR1 gene that can expand. And when it expands over a certain critical length, it causes Fragile X syndrome. And we see here an individual, a cell line network sequence that has 1 wild type FMR1 allele and 1 expanded FMR1 allele. So that the mechanism of pathogenicity, or what's thought to be the mechanism of pathogenicity for this repeat expansion, is silencing by methylation upon expansion. And that's you can see that clearly here. If you look at the short alleles, you can see very few of them are methylated, which is shown in pink. But when you have the expanded alleles, they are all heavily methylated. So here with HiFi reads, you're able to read through these repeat expansions, see their full sequence. You can see CGGs when there's AGG interruptions, and then you can see the methylation status with the expanded allele being highly methylated, and some of the short alleles being methylated as well, but it being much less prevalent. Another interesting example of methylation in a repeat expansion is from Tomi Koskinen at Children's Mercy Kansas City. He had sequenced an individual that has myotonic dystrophy due to a DMPK repeat expansion with HiFi. And you can see here on the left, looking at the genetics of that individual. You see that there's 2 alleles present. You can phase the reads by the single nucleotide variation. And on the top allele, you see that there's a large insertion, a mosaic insertion. So there's much different length across the reads. But you see that there's a large insertion that corresponds to a DMPK repeat expansion. And then when you look at the corresponding epigenetic profile, you can see that this expansion induces hypermethylation directly adjacent to the expansion. So the wild type allele is hypomethylated, is blue in this window, and the expanded allele is red. Tomi actually this week they released a fairly large set, a preprint describing a fairly large set of Firebase HiFi genome sequencing, looking, so it includes this one case that I just described, but it also includes many more individuals and looking at methylation and many more interesting aspects of phase methylomes. So they provide direct haplotype resolved Firebase HiFi genome sequencing of 276 samples from 152 families. And from that, they identify a number of interesting novel characteristics of the impact of genetics on epigenetics and epigenetics on gene activity. This is just showing one example where they have identified a structural variation, in this case, a 69 base pair deletion, that induces hypermethylation adjacent to that deletion. So this is showing an individual, the proband, where you have one allele showing not much methylation, the other allele showing high methylation linked to the 69 base pair deletion. And if you look in the mother, you can see that the methylation status is the same in the mother. And you can look across trios and see that this variant is associated with this nearby methylation. So you get this direct phasing of these structure variants and methylation in a single assay. They also were able to use this approach to identify a previously overlooked repeat expansion in the DIP2B gene that was causative of disease in one individual. Okay, so to summarize the key benefits of Firebase Hi-Fi sequencing, that it provides a genome-wide measure of methylation, all from a single library in a single run. And because it comes with PacBio Hi-Fi reads, you get all the other benefits of that sequencing. You get phasing, uniform coverage of the genome, and high mapability. Just to end, I want to point to some resources and data sets that might be interesting. So there is we've made available Firebase sequencing data set of HG002. That's available at this location on the PacBio cloud. This is the data set I use for many of the examples. There are also a large number of Firebase sequencing data sets available from the Human Pangenome Reference Consortium, but this is one available from PacBio. The analysis software, the Primrose and the PBCPG tools, are both available on PacBio's GitHub. So github.com/Pacific Biosciences. The Primrose software is the one that also runs on the Sequel IIe systems now. So those of you who are running Sequel IIe systems will just get this directly off the instrument. But those who would like to do analysis off instrument can download the software from GitHub. And then finally, I mentioned earlier that IGV starting in version 2.10 provides visualization of these marks, and it's improved further in version 2.14. So I would recommend using a new version of IGV, version 2.14 or newer. And in this version of IGV, when you have an alignment track, you can right-click on that track and choose the color alignments by. And if you choose base modification 5MC, then you'll get this style view where the MM and ML tags encoded in the BAM are used to color bases, either blue if they're very unlikely to be methylated, or red if they're very likely to be methylated. You can also get some more information on Firebase sequencing on PacBio's website. So if you go to pacb.com/epigenetics, you will see some description on Firebase sequencing, and also you can look at some brochures that link to many of these resources as well.
Well, thank you, Aaron. I think to me this is one of those kind of revolutionary things, because it's really, opening up something that it's not that it wasn't always accessible before, but it was hard. Now it's just really simplified the process.
Of course, thanks everyone thanks for the invitation, and thanks for everyone for joining. And of course, they can reach out to me or Matt or PacBio support to help with any questions you have on Firebase sequencing or SMRT Link.